These organisms have no brain, no neurons, and no decision-making apparatus of any kind. Several of them have demonstrably outperformed human engineering. This is not a list of primitive survival. One is one of the oldest and largest livings on Earth. The Blob. Decades of human engineering and supercomputing were required to design the Tokyo subway system. In 2010, a slime mold replicated the exact layout with mathematically superior resilience in 26 hours. No neurons, no brain. Physarum polycephalum inhabits forest floors, urban soil, and rotting wood on every continent. And it has been quietly solving optimization problems since before the first vertebrate existed. It is a biological calculation engine driven by pure hydrostatic pulsation. Every 60 seconds, actin-myosin filaments violently contract, pushing cytoplasm through tubular veins at high pressure. It does not think. It pumps itself forward. Spatial memory. In humans, a dedicated brain structure requiring years of neurological development. Physarum solved with residue. It excretes a glycoprotein slime trail. Contact with its own excretion shifts the osmotic gradient, forcing an immediate vector change. The memory is not stored internally. It is deposited on the floor and abandoned. A cognitive function replaced by a chemical stain. Upon finding resources, it prunes inefficient veins to maximize fluid dynamics. An autonomous pipeline. Its only record of its own history is the debris it left behind. And while this single cell optimizes microscopic mazes, deep underground, a subterranean anomaly is already generating enough hydrostatic force to fracture the bedrock of entire continents. The Social Amoeba. The food is gone. The bacterial population has collapsed to zero. For Dictyostelium discoideum, a single-celled organism with no nervous system and no mechanism for awareness of any kind, this is a death sentence. Unless the dead start broadcasting. Dying cells secrete cyclic adenosine monophosphate. Neighbors register it, migrate toward the signal, and immediately rebroadcast it themselves. The chain reaction propagates exponentially until up to 100,000 independent cells have locked together into a single crawling mass, moving as one unit toward the surface. What follows has no equivalent in any coordinated animal behavior. The leading 20% undergo terminal vacuolization, digesting themselves from the inside out to form a rigid cellulose stalk. A structural scaffold the survivors climb to disperse their spores from elevation. No orders were issued. No individual cell processed the logic of its own death. 100,000 autonomous organisms with no brain between them executed a synchronized sacrifice with perfect compliance. The Portuguese Man o' War. Physalia physalis is not a jellyfish. It is a siphonophore, a colony of clones, each physically locked into a single function. One forms the gas bladder. Others are exclusively toxic harpoon arrays, digestive tracts, or reproductive units. No clone performs more than one role. No clone directs any other. There is no coordination because there is no coordinator. The 30-meter tentacles fire in nanoseconds on mechanical contact alone, completely independent of the rest of the collective. No signal required, no permission granted. This is the consequence. There is no off switch. A tentacle severed from the main organism washes onto a beach. The organism it belonged to is dead. Weeks have passed. A human makes incidental contact. The mechanoreceptors, with nothing to consult and no instruction to stand down, execute the full kill sequence, injecting neurotoxins that trigger cardiovascular collapse and respiratory failure. The weapon had no idea the war was over. It never had any idea about anything. The red-beard sponge. Microciona prolifera is forced through a fine silk sieve. Every physical connection is severed. The organism is reduced to thousands of isolated cells suspended in open water. This is not fatal. It is a temporary mechanical inconvenience. The isolated cells, archaeocytes, immediately execute cellular totipotency. They drift through the water column, identifying each other through specialized glycoproteins called aggregation factors. The chemical adhesion is precise enough to distinguish self from non-self at the molecular level. The proof? Pulverize two completely different sponge species and combine their cells in the same water column. No brain. No sorting mechanism beyond surface chemistry. Within hours, the cells have blindly separated back into two genetically pure organisms. Zero cross-contamination. Within 24 hours, the original sponge has fully reconstructed its pumping infrastructure from scattered debris. Not wound healing, a complete rebuild from biological dust. Destruction, for this organism, is simply the starting condition of reconstruction. The Upside-down Jellyfish. Cassiopea xamachana never moves. It rests inverted on the shallow sea floor, absorbing sunlight to fuel its symbiotic algae. A stationary organism with no brain, no locomotion, and no capacity to pursue anything, but it controls the space above it completely. Upon detecting water displacement, it secretes a dense mucus cloud loaded with cassiopea, hollow cell clusters armed with thousands of nematocysts, covered in hair-like cilia. Once released, these detached clusters actively rotate and navigate through the water column, autonomous, self-propelled, entirely independent of the organism that produced them. When a cassiopea strikes a target, the mechanoreceptors fire automatically. Neurotoxins paralyze the prey. Digestive enzymes begin external liquefaction before the nutrient package sinks back to the seafloor. For divers, the result is stinging water syndrome, pain and dermatitis from an enemy never physically touched, deployed by an organism that never left the bottom. Total immobility, but complete territorial control, and no brain required. The Humongous Fungus. A single organism currently occupies 9 square kilometers of forest in Oregon. It weighs an estimated 35,000 metric tons. It has been expanding for somewhere between 2,000 and 8,000 years. It has no brain, no central coordination point, and no mechanism for awareness of its own scale. The fungus is alive and growing actively, destroying every tree around. The visible mushrooms are irrelevant, temporary exhaust vents for the organism below. Beneath the soil, the mycelium aggregates into thick, melanized cables called rhizomorphs. Through osmotic water intake, these subterranean structures generate massive internal hydrostatic pressure. At the apex, that pressure is focused onto a microscopic contact point. The fungus cracks open healthy tree bark through sheer hydraulic force. Not chemical erosion, not slow infiltration, mechanical fracture, the same principle as industrial hydraulic drilling. The moment the bark splits, toxic enzymes flood the breach, dissolving the wood from within. Across 9 square kilometers, the network simultaneously registers nutrient gradients, redirects water, carbon, and phosphorus through a fluid dynamic grid, and positions new hydraulic drill heads against the next target. A continental-scale mining operation running continuously, without a single moment of decision, in complete darkness. The largest organism on Earth. Not a whale, not a tree, a fungus that has spent millennia quietly fracturing the bedrock of a forest and has never once been aware that it exists.